So here we're going to walk through a primer on the nerve and muscle subject of neuroanatomy. We're going to walk through histology, biology, and physiology. We'll start with peripheral nerve histology. We'll break it down into its connective tissue and nerve itself. So from a connective tissue standpoint, the terms we'll look at are epineurium. This is the most superficial outer covering of the nerve. And then perineurium, which encases separate nerve fascicles. And then endoneurium, which is this loose connective tissue internal to the perineurium. Next, we're going to look at the nerve itself. This is basically what we define as a peripheral nerve in this illustration. This is a nerve fascicle surrounded by perineurium. These are nerve fibers, the axons. And then this is the myelin sheath here, which surrounds the peripheral nerve axon. And then the Schwann cell, which produces the myelin sheath, and we can see that illustrated here. Nerves that are heavily myelinated travel the fastest. Nerves that are unmyelinated or poorly myelinated travel the slowest. In terms of sensory nerves, nerves that encode proprioception, joint position, two-point discrimination, these are the large fibers. And then nerves that encode itch, temperature, or pain, these are small, poorly myelinated fibers. Eukaryotic cell architecture, we see this tutorial in a lot of subjects throughout the site, and that's because it's so core to biology. So this is the cell itself. These are the key organelle structures here, and then these are their key functions, and you can walk through this tutorial for a good review. Neuron cell structure. So two classification systems that we'll introduce, a functional and then a physical. From a functional classification standpoint, sensory neurons accept afferent sensory information, interneurons relay information, and then motor neurons emit efferent motor commands. From a physical classification, we're talking about different poles here. So a neuron most commonly will be multipolar. It has 3 or more processes on the cell body itself. And it alternatively can be bipolar, where there's 2 processes emanating from the cell body itself. It can be pseudo-unipolar, meaning that there's really only one pole, but it quickly branches. And the dorsal root ganglion, the sensory neuron, is a great example of this. And then finally, it can be unipolar, where there's a single axon emanating from the cell body here. So this is a cell body. We're going to walk through some key terminology over here. This is the neuronal command center. It contains those eukaryotic cell organelles. There are dendrites. These are processes that receive these signals, and they're attached to the cell body here. And then there's the axon here, which is transmitting, in this case, the signal itself. And then we see the myelin sheath wrapped around the axon. These are these nodes of Ranvier. There's this concept of saltatory conduction, where the conduction skips between these different nodes. Remember or know that the myelin sheath in the peripheral nervous system is produced by Schwann cells, and the central nervous system is produced by oligodendrocytes. That's a key distinction. This is this junction here, this synapse, this this terminal bouton, this axon terminal, or also known as the synaptic knob. It's the terminal at the distal end of the axon. We'll see later how these neurotransmitters are dumped into the synaptic cleft, and then they're picked up by this postsynaptic cell. Glial cells, these are white matter cells. So over here, we're going to demarcate about central nervous system glial cells versus peripheral nervous system cells. And we'll denote that most of the ones that we're going to look at are macroglia. Starting with this astrocyte, these are its key physiological functions. We see it here. It has numerous processes. Microglia are represented here. They phagocytize pathogens and dead neurons. Ependymal cells, they form this permeable barrier, and we can see that this ventricle has cerebrospinal fluid, and they have these cilia, and they're key to circulating CSF. So this is a key histological finding, the cilia on the ependymal cells to circulate the CSF. Oligodendrocytes, we previously referenced, they create myelin sheath in the central nervous system. They have a similar shape to the astrocytes, but fewer processes, and you can see the comparison right here. When it comes to the peripheral nervous system here, we comment on two key cells. These are the satellite cells. We can think of them like the astrocytes of the peripheral nervous system. And then there's the Schwann cells, and we've seen these already before. They create myelin sheath. Again, here's the node of Ranvier, these gaps in between myelin to help with saltatory conduction. Again, these are the peripheral nervous system counterpart to the oligodendrocytes of the central nervous system. Neuronal excitation. So we're going to take a deep dive into this tutorial here because it summarizes many of the tutorials that follow in this subject. So neuronal excitation, these are two key concepts we're going to walk through: electrochemical equilibrium and then the equilibrium potential. So we're going to look at sodium and potassium. The way to first think about this is to think of our cells as just a single cell growing up in salt water. So when we look at blood levels of sodium, the blood level is quite high when it comes to sodium, 140 milliequivalents per liter, versus, as we'll see with potassium, the blood level is quite low. It's maybe 4 milliequivalents per liter. So surrounding the cell is this heavily salt fluid. Inside the cell, there's a low amount of salt. So what wants to happen is salt wants to move from outside of the cell to inside of the cell. And it does that through this sodium leak channel here. So that's the chemical gradient. Now, when the sodium rushes into the cell, it creates additional positive charge within the cell. This needs to be counterbalanced by an electrostatic gradient going the other way. So ultimately, the membrane, the equilibrium potential for sodium is plus, and these are rough estimates because they vary, 60 millivolts, because additional sodium brings in positive charge. Now it's important to remember that like charges repel themselves. That's why it's counterbalanced by this electrostatic gradient going the other way. So there's this electrochemical equilibrium because there's two components to it. One is chemical and one is electrical. From a potassium standpoint, as we mentioned, there's high levels of potassium within the cell and there's low levels of potassium outside of the cell. You'll notice that all these ions are counterbalanced by a chloride ion, which is negatively charged. So similar phenomenon, only the opposite directionality. So potassium here moves from inside the cell to outside of the cell through a leak channel, and the electrostatic gradient then is in the opposite direction as well. Positive charge is moving out, so positive charge has to move in. And ultimately, the equilibrium potential for this ion is roughly negative 90 millivolts. So here's a negative charge and then a positive charge. So now we're going to put all of this together in a neuron because there are multiple different ions within a single neuron. So the membrane potential is the voltage potential of the intracellular space in reference to the extracellular space, and it combines all of the different charges. So what are those charges? As a review, sodium, high levels outside of the cell, it's moving into the cell. Potassium, high levels within the cell, it's moving out of the cell. And both of these have these opposite directionality electrostatic gradients. And then there's this sodium-potassium ATPase pump. Now, it requires ATP, it requires energy, because it's pushing chemicals against their gradients. So sodium is being pushed out and potassium is being pushed in to counterbalance the natural directionality of these chemicals. Overall, the membrane potential for the neuron is negative 70 millivolts. So we introduce chloride. Chloride has roughly, we'll call it for our purposes here, negative 80 millivolt equilibrium potential. So then there is this electrochemical driving force, which is the drive, the push that's happening based on the difference between the equilibrium potential of the ion and the overall membrane potential itself. And these concepts are further elaborated upon in the tutorial itself. So let's talk about neuronal signaling now. So a neuron, a presynaptic neuron here, postsynaptic neuron here, there's an action potential along the presynaptic neuron. It induces this calcium influx here. For this purpose that we're using a calcium activated channel. There's a vesicle release of neurotransmitter which binds to this postsynaptic ligand-gated sodium channel. Sodium rushes in, causes depolarization, and then voltage-gated sodium channels along the postsynaptic membrane are activated, and they stimulate this action potential to move along the postsynaptic cell. So let's get into the action potential itself to understand how this is activating action potentials through depolarization. So at baseline, there's a resting state where the voltage-gated ion channels are closed and potassium leak channels are the driving force. So if potassium is leaking out of the cell, then the cell is becoming more negative in its resting state. When there's depolarization, ligand-gated sodium channels open, as we saw here. What does that do? That increases the membrane potential. It becomes less negative, more positive. When it reaches a certain state, and we call that here at negative 55 millivolts, then there's rapid opening of these channels, and there's a sudden spike, and that's the action potential itself. At some point, this action potential has to end, and when it does, there's repolarization. The sodium channels are inactivated now. The sodium can no longer rush into the cell, and then the potassium channels open, and they move out of the cell to bring things back to the resting state membrane potential. Ultimately, there's this hyperpolarization state where the potassium channels are closed. The sodium channels are closed. The potassium channels remain open, and the membrane potential is actually lower than its standard resting state. And there's this inactivation period where the cell can no longer be activated. So all of these concepts are further elaborated upon in various tutorials in this subject. The first is gated ion channels. Another is more information about the resting membrane potential. More information about the action potential itself, and then neuronal signaling. And this brings up this concept of heavy myelination versus light or unmyelination. For instance, there's these group A fibers that are the largest diameter, thickly myelinated. They travel up to 120 meters per second, versus group C, smallest diameter, non-myelinated. They travel at roughly 1 meter per second or less. So if you think about when you step on something that you know is going to hurt, there's this delay in the pain. You feel it because these large fibers pick up the sensation itself, but you know the pain's coming, but it takes a while to get there because the smallest diameter fibers carry the pain impulse itself. That's where the disconnect comes from. And we talked about, and this is covered here, but these nodes of Ranvier and saltatory conduction, and we get into those details in this neuronal signaling tutorial. There's also this postsynaptic integration tutorial, and I just want to point out these excitatory versus inhibitory activations, EPSPs versus IPSPs, and then there's a summation. So that tutorial gets into those details. And then there's information on various neurotransmitters. Okay, so moving on to muscle excitation physiology, we start with muscle excitation and contraction coupling. So we're coupling the excitation of muscle and then the contraction physiology itself. So a muscle cell contains myofibrils. Myofibrils are made up of myofilaments, and these are the various myofilament components. There's actin, myosin, and titin. We don't focus on titin, we only focus on actin and myosin. This is our representative myosin with its neck and its head here and its tail. And then here is actin with myosin binding sites on it. It also contains tropomyosin and troponin, which has calcium binding sites. So let's see how this works. So their acetylcholine is released from the presynaptic cell. We saw how neurotransmitters get released and then picked up at the postsynaptic cell, and that's what's happening here. There's activation of this action potential along the postsynaptic membrane. The action potential then activates release of calcium into the cytosol. The calcium then binds to troponin, which causes a shift in tropomyosin, and that's what's represented here. And then when there's a shift in tropomyosin, myosin is able to bind to the myosin binding site on actin, represented here. The contraction steps themselves, ATP is present, and there's an unbinding here. Then when there's hydrolysis, there's a rotation of the myosin head, and now we have high energy ADP. The myosin head then binds to actin here, and there's a release of phosphorus or phosphate, and then there's this power stroke. And you can see that, and then this is this activation cycle here. And you can see more details of that, and then also get into the structure of a sarcomere with the A band, H zone, and I bands. And this is very important information to know cold here, but you can see those details of contraction further elaborated on this tutorial. The final tutorial I want to introduce is the motor unit itself. There's an innervation ratio, which is very important. It connotes whether or not there's going to be gross motor control or fine motor control. For instance, extraocular muscles have very few muscle fibers for every motor neuron, whereas muscles of the deltoid have a large number of muscle fibers for every neuron. So there's a single motor neuron, multiple axon branches, and multiple muscle fibers. That's represented here. Here's the neuron itself, here's the axon with numerous branches, and then here are the muscle fibers. In terms of muscle fiber types, again, important information to know cold. There's slow contraction, which are red because they have high levels of myoglobin because they perform aerobic physiology. They rely on ATP. And then there's fast contraction, which are white because they have low levels of myoglobin and they have anaerobic physiology. And we see that laid out here. So slow, red, aerobic, high levels of myoglobin, and then they have the highest level of fatigue resistance. So they're resistant to fatigue. On the contrast, the white fibers, anaerobic physiology, fast twitch, and they are have low fatigue resistance, meaning that they fatigue easily. And then there's an intermediate type, which is a blend of both. So that is a whirlwind of nerve and muscle histology, biology, and physiology. Again, there are tutorials on the site that go into all of these concepts in greater depth, but I hope that helps you with some core concepts of neuromuscle physiology.